Hello, my name is Kostas Pitris. I'm a professor at the Department of Electrical Engineering. Welcome to the Optical Diagnostics Lab. In this lab, we have the equipment and we develop technologies for optical diagnostics, which means that we use laser light to develop methodologies for the diagnosis of various diseases. Our main research areas consist of optical coherence tomography and Raman spectroscopy. In optical coherence tomography, we both develop novel technologies to perform optical tomographic image of tissue microstructure, as well as develop new algorithms which allows us to detect neoplasias in early cancer. These technologies are based on new swept scan sources and also liquid crystal phase filters, which allows us to manipulate the beam and create new shapes and improve the resolution. Our group is actually the leaders in extracting novel information from optical coherence tomography. The other set of projects has to do with Raman spectroscopy, which is another type of an optical diagnostic. It's a spectroscopic technique which provides biochemical information regarding the sample under investigation. We use it to identify bacterial infections in urine to diagnose urinary tract infections, and our group was the leader in developing methodologies to determine the antibiotic sensitivity of the bacteria. In this lab, we have 3 graduate students, 1 postdoctoral student, and 4 undergraduate students on several aspects of the project. Not all of them work on the hardware, some work on the analysis algorithms and artificial intelligence, which we combine with the data that we capture to create the novel diagnostics. This is an optical coherence tomography system, which is very similar to ultrasound in the sense that it measures waves reflected back from tissues. Unlike ultrasound, it uses laser light to detect the tissue microstructure, so the resolution is about 10 to 100 times better than ultrasound. So, for example, in this image you can see the skin of my finger, where you can see the epidermis and the endodermis at great detail. We use this system in a project to diagnose early stages of radiation dermatitis to be able to then guide treatment and monitor therapy. This is the small animal molecular imaging lab. This is where we perform experiments on the response and nature of cancers in small animals, which allows us to experiment with novel nanotechnology-based sensing and therapy. The lab is equipped with a system that combines optical molecular imaging, fluorescence tomography, and a CT scanner to image in 3 dimensions small animals. This is a state-of-the-art equipment that is actually quite expensive, but allows us to do many interesting experiments in vivo with the animals anesthetized in the system, and we can watch or we can image the anatomy of the animal using the X-ray CT, and also do molecular imaging to see optical response of the cancer itself, as well as the pharmaceuticals that we are injecting, using the optical part of the imaging, which can perform both regular fluorescence imaging in 2 dimensions as well as 3-dimensional fluorescence tomography. In this facility, we do not breed the animals, but we do have the equipment to house the animals in a sterile fashion with individually ventilated cages in a dedicated changing station. These are HEPA filtered, so we can keep immunocompromised animals here without actually exposing them to any harmful agents. Part of the work done here is to actually implant the animals with tumor organoids from human cancer cells. We construct those on scaffolds made of nanofibers, polymer and multiple polymer nanofibers, which can be created with the system, the electrospinning system shown there, which is it's a system that actually injects small quantities of a material towards a cathode with a very high voltage difference. And in the process, it gets stretched nano dimensions. It creates a nano scaffold onto which we grow the